I'm Harley Smith, and this is the Master Growers course. Today we're going to be taking a closer look at microbial inoculants and compost teas. Plants and microorganisms have co-evolved on the planet, not just to help each other survive, but to help each other thrive. They've worked together in a mutually beneficial way as they've evolved on the planet. Plants are autotrophic. That means they're self-feeders. They make their own food. Plants take in carbon dioxide through the leaves, they take up water through the roots, and then in the presence of full spectrum light, they do photosynthesis. They make carbohydrates, photosynthates. Some of those carbohydrates they use for energy to grow and reproduce. Some they store in the flowers, in the fruits, in the seeds, in the roots. But some they leak from the roots to feed the plant growth-promoting rhizobacteria and the mycorrhizal fungi right in the rhizosphere. And in exchange for those carbohydrates, the microorganisms make biologically active molecules that aid with the uptake of water and minerals. And those are what we call the biostimulants. But today we're going to be looking at the biostimulant factories. We're going to be looking at the superstars of the rhizosphere. And the first time in the history of agriculture, we're able to harvest microorganisms from the most fertile soils from around the world, harvest other microorganisms from disease-suppressive soils, and then combine them in a way that they work with each other. They work with each other instead of compete with one another. We can do things that have never been done in the history of agriculture. We can find ways now, there are microbial blends coming out of the universities. They're able to cut fertilizer usage in half, reduce the nitrogen fertilizer, but increase quality and yield. We found ways to lower, reduce, or even eliminate the need for pesticides and fungicides. And we've learned how to improve quality and improve yield. So this is the new frontier, and it's very important for the future of agriculture. So today we're going to look at some of those superstars from the rhizosphere. Let's start with the mycorrhizae, the mycorrhizal fungi. Myco means fungus, rhizae means root. So mycorrhizae literally means fungus roots. They're beneficial fungi that infect the roots in a beneficial way. They penetrate the root cells, and then as the mycelium, the threads, grow in the rhizosphere, they harvest and mine nutrients. They unlock phosphorus from the soil, they feed it to the plant. So mycorrhizal fungi will increase the surface area of the root zone that plants have by hundreds or even thousands of times. That improves the uptake of water, improves the uptake of nutrients, makes them more drought resistant, and even stimulates root growth. Remember too, the more efficiently plants take up water and minerals, the more chemical reactions they can do, the higher the Brix. Brix is the measurement of the sugar content in the sap. So one of the goals of all of the Master Growers classes is to grow high Brix plants. If we have more sugar production by the plant, there's more stored energy that can be released in abundance of fruit and flowers. There's more stored energy that can be used to fight off pests and disease or to adapt themselves to environmental stress. And there's more stored energy that can be used to take up essential elements for plant growth and reproduction. So growing high Brix plants is the key, but mycorrhizal fungi aid with the production of the sugars by the plant. But one of the main benefits of mycorrhizal fungi is that they unlock phosphorus from the soil. Now phosphorus gets locked up in the soil very easily. It locks up with calcium to make calcium phosphate. That's what your bones are made out of. 95% water insoluble. And if it's insoluble, it's unavailable to the plant. Also, phosphorus locks up with iron to make iron phosphate. It makes the iron unavailable to the plant. So, if there's a phosphorus deficiency in infertile soil, plants will exude signal molecules to attract the mycorrhizal fungi. It infects the roots, and as the mycelium grows through the root zone, it exudes organic acids, it exudes enzymes that unlock phosphorus from the soil, unlock phosphorus from organic matter, and feed it to the plant. So that's one of your first choices you have to make as a grower. Am I going to be doing all organics, or am I going to do mineral nutrition? If you're going to do organics with rock phosphate and bone meal, then you definitely want to inoculate the roots. Very first thing, right at transplant, with the mycorrhizal fungi. It'll aid with the uptake of the phosphorus. Now the downside is it costs an energy budget for the plant to feed the fungus. In other words, over 20 or to 30% of the energy of photosynthesis is leaked from the roots just to feed the mycorrhizal fungi. So, if there's plenty of water-soluble phosphorus, the plants will treat the mycorrhizal fungi as a pathogen. Instead of attracting them to the roots, they'll make enzymes to repel the mycorrhizal fungi. So, if you're growing with all mineral nutrition, the mycorrhizal fungi won't have nearly the benefits as it would in organic fertilization. So mycorrhizal fungi are especially important with organic nutrition or in a starter fertilizer to help unlock phosphorus from the soil and feed it to the microorganisms. Now the next on our list are the plant growth promoting rhizobacteria. Unlike the fungi, they don't spread out in the root zone, they colonize the surface of the roots. And the plant growth promoting rhizobacteria are the biostimulant factories. They feed on the waste materials from the plants, from the carbohydrate exudates and amino acids, and they change them into biostimulants. Microorganisms will actually make growth hormones, IAA, right on the surface of the roots. Now, why would a microorganism want to stimulate more roots from the plant? That's because the genetic code in every living cell and every living thing says the same thing. Be fruitful and multiply. So if the microorganisms, if the bacteria stimulate root growth, there's more surface area for the microorganisms to colonize. Also, if there's more surface area, there'll be more leaked carbohydrates to feed the microbes. So it's a competitive advantage for the microorganisms to stimulate root growth. Microorganisms such as the plant growth promoting rhizobacteria also produce B vitamins. Now a high dose of B1 right on the roots stimulates cellular metabolism of the microorganism themselves and the other microorganisms in the root zone. And at the same time, the B vitamins stimulate the systemic induced response of the plant. It activates the immune system so that if the plant is under attack from a pathogenic microorganism such as Pythium or Rhizoctonia or one of the root rots, the plant's natural immune system responds almost immediately. Why would a microorganism stimulate the immune system of the plant? To protect its food source. They're working together in a mutually beneficial way. Also, microorganisms make digestive enzymes right on the surface of the roots. Now, plants can't take up large organic molecules. Those large molecules have to be broken down into smaller and smaller particles until they're in their ionic form. That's the form of plants actually take them up through the roots. So microorganisms make the digestive enzymes to make the minerals more available to the plant. It's called mineralization. Protease, it breaks down proteins, some of which are just slough from the cells of the plant as the roots are growing through the soil. It breaks down the proteins into amino acids. Now, two of those amino acids, glutamic acid and glycine, are intermediate chelators. Chela means claw. So a chelate is an organic molecule that attaches to a mineral like a claw. It holds it tightly enough so that it doesn't lock up with other minerals in the soil, but loosely enough so it's available to the plant on demand. Microorganisms also make organic acids. As they feed on the sugars, they make what are called Krebs cycle intermediates. The Krebs cycle is cellular metabolism, respiration, to change the carbohydrates into energy. But in the process, they make organic acids, such as citric acid and malic acid. And those organic acids lower the pH in the root zone. The surface of the roots could be 2 points lower in pH than the surrounding soil, improving the uptake of the transition metals, such as iron, copper, manganese, and zinc by the plant. Also, some microorganisms, like Bacillus subtilis, GBO3, make a special chelator called a siderophore. Siderophore literally means iron carrier. It's one of the best chelators of iron in nature, so it keeps the iron soluble and available to the plant. And finally, some microorganisms make volatile organic compounds, a bouquet of aromas. So even if the microorganism doesn't touch the plant, doesn't even touch the root, it makes a bouquet of aromas that can stimulate up to 600 genes in the plant. One of those genes tells the plant, take up more iron. So not only do microorganisms make the iron soluble, make it available, they also tell the plant to take up more of it. As the plant takes up iron, it stimulates the plant to make more chlorophyll. Makes more chlorophyll, harvests more light energy. Harvests more light energy, makes more sugars. Makes more sugars, some of them are used for energy to grow, to reproduce, to make more roots. Some of them are stored in the flowers, in the fruit, for the next generation. Some are leaked from the roots as exudates to feed the plant growth promoting rhizobacteria. So the cycle is complete. The plant feeds the microorganisms, and the microorganisms in turn help feed the plant. There are two main ways to add soil biology to the root zone, either microbial inoculants or compost teas. Now, the secret of a high-quality compost tea is high-quality compost. You want to have compost from a variety of sources. You want the compost from farmyard waste and also from worm castings. Now, just because there's a compost in your soilless mix or in your soil, it doesn't mean that the microbiology is rich in that compost. A lot of the composts that are made commercially are produced in large windrows. So the center of the compost gets very, very hot, and it will kill some of the mesophiles. Those are the microorganisms that grow at medium temperatures. Some of those mesophiles are some of the superstars of the rhizosphere, especially for stimulating the immune system of the plant. The actomycetes are one of those that are mesophiles. Also the trichodermas, very powerful microbes in the rhizosphere, one of the main superstars, but they grow at moderate temperatures. So even if you have a potting soil with compost in it, it will behoove you to add microbial inoculants to that soil. The compost tea will add diversity, wide diversity of microorganisms, and it also adds some fertilizer value. As the microorganisms are digesting the compost, not only are they making the minerals available, they're manufacturing some of the biostimulants that we just talked about. So compost tea goes way beyond just the benefits of the mineral nutrition alone. It's stimulating the uptake of those nutrients as well. Now, the downside of compost tea is that it has to be actively aerated. The aerobic microorganisms are the good guys. The anaerobic fungi are the bad guys. The anaerobic fungi grow in stagnant water without oxygen. Those are the root rots, the rhizoctonia, the pythium. They attack the roots, break them down. They're pathogenic to the crop. And that makes it so the plant can't take up minerals efficiently. You'll see symptoms of nutrient deficiencies in your plant on the top growth because the roots are unhealthy. But microorganisms protect the root cells and actually make antifungal and antibacterial agents. But it has to be done in actively aerated compost tea. So you want to make sure that it's continued to have a bubbler in there or that the water is circulating as you're brewing your compost teas. The other way to add soil microbiology is microbial inoculants. Now, the secret of a good microbial inoculant blend is choosing microorganisms that complement one another instead of compete with one another. In a compost tea, you're going to have wide diversity of microorganisms. Some will work together, some won't. But in a microbial inoculant, you can pick just the superstars of the rhizosphere, those with superior characteristics. I work with one microbiologist out east. He has 1,200 strains, pure strains of plant growth promoting rhizobacteria in the lab. And out of that 1,200 strains, he can combine the ones that are the best of the best at what they do. Some are superstars at taking nitrogen out of the air, changing it into fertilizer from the plant. Other specific strains are better at unlocking phosphorus. Others are better at making the root growth hormones or the other growth hormones for the plant to stimulate root growth. Others are a little bit better at making plant protection agents or rescuing the plant from stress. So if you have 1,200 strains harvested from the most fertile soils and from disease suppressive soils, you can pick those that just have the genetic superiority in their characteristics and then combine them in a way so it's a dream team to inoculate the rhizosphere. Another benefit of the microbial inoculants is that they have a guaranteed analysis. Now, it's not only saying what's there, but also what isn't there. They're grown in the lab under controlled conditions with quality control. In a compost tea, it can be very, very beneficial. And some of you are masters at making your own blends. It's a science and an art. But this is more of the science. If you have an improperly made compost, there could be pathogens in there. There could be E. coli. There could be salmonella that cause human diseases. Just because it's organic doesn't mean it's good. There's good guys and there's bad guys in the soil and also in compost. But in a microbial inoculant, you won't have any of those human pathogens or pathogens for the plants. If you look at the label, you can see what the guaranteed analysis is. Each of the microorganisms will be put on the label as CFUs. CFU stands for Colony Forming Unit. To be a CFU, of course, the microbes have to be alive, and they have to be able to form whole colonies. If you look at a petri dish, you see the discs that are on there, those are colonies. So that's how we test it. It has to be guaranteed amount. Another benefit of microbial inoculants is you can put together specific blends for specific purposes. You could put together a blend that's dominant in the mycorrhizal fungi, especially if you're going to be doing all organics. On the other hand, you could put together a bacterial-dominated blend, even blends for specific stages of the plant's growth. You could have one blend that just colonizes the roots early on to improve the uptake of phosphorus, improve the root strike and establishment of the plant. You could put together another blend for the bloom stage that would have more of the microorganisms that produce plant protection agents or rescue the plant from stress or aid with the finishing power of your plants. Or you could put together a blend that's specifically designed for disease protection to help the plant recover from powdery mildew or from other pathogens in the root zone, because some microbes are superstars at making plant protection agents. So let's take a minute to look at some of the key players in the microbial dream team. We'll start with the phosphorus solubilizing bacteria. Phosphorus gets locked up easily in the soil. We talked about that. It locks up with phosphates, it locks up with iron, makes nutrients unavailable to the plant. But some microorganisms produce organic acids and enzymes such as phytase and phosphatase that unlock phosphorus from the soil, unlock phosphorus from organic matter, feed it to the plant. Now, some phosphorus solubilizing bacteria are synergistic with the mycorrhizal fungi. Remember, the mycorrhizal fungi inoculate the roots, and then as they grow in the rhizosphere, they exude organic acids and enzymes to unlock phosphorus. Now, the plant growth promoting rhizobacteria, the phosphorus solubilizers, hitch a ride on the mycorrhizal fungi. It takes them to places they could never have reached on their own in the soil area around the rhizosphere. On the other hand, they swim in the soil solution to places even the mycorrhizal fungi can't reach. They unlock phosphorus from the soil, feed it to the fungi. The fungi, in turn, feeds it to the plant in the roots. So they're synergistic. If you have a starter blend with mycorrhizal fungi, make sure that it includes phosphorus solubilizing bacteria. One of the best solubilizers of phosphorus in the rhizosphere is a bacteria called Bacillus megaterium. You definitely want that one on your team. It's a giant in the microbial world. It also forms chains, massive enzyme producers, and it's excellent at unlocking phosphorus. Next come the nitrogen-fixing bacteria. Some microorganisms are able to harvest nitrogen from the air and change it into fertilizer for the plant. Now, with nitrogen fertilizers, they're made the same way, but we have to take in the nitrogen from the air under high temperature and high pressure in order to make those fertilizers, and they're extremely soluble. For example, nitrate nitrogen, you can feed it to the plant. If there's a big rain, it'll wash the nitrogen away from the root zone, and it could go into the groundwater and into surface water. But if you use the nitrogen-fixing bacteria, they're fixing nitrogen, they're manufacturing fertilizer right on the surface of the roots. It's 95% efficient. So some of those microorganisms work in a team. Some fix the nitrogen from the atmosphere and change it into ammonium. Other microorganisms change ammonium into nitrite. Others change nitrite into nitrate, and then the plant takes up the nitrate nitrogen. So we're sequestering nitrogen in the root zone and manufacturing it real time, right when the plant needs it the most. Another thing to remember is that microorganisms such as bacteria rarely die of old age. They're eaten by other microorganisms like protozoa. A protozoa eats a bacteria. It uses some of the nitrogen for its own use, for growth and metabolism, but then it spits out the excess. That excess nitrogen is fertilizer for the plant. So we're sequestering and recycling nitrogen directly in the root zone where it's needed the most. Some of the microbial blends coming out of the universities, they found that they could reduce the nitrogen fertilizer in half, by 50% or more, and increase yields at the same time. That's revolutionary for our planet, not just to protect the environment from nitrogen runoff, but to help the farmers to be more efficient, to restore the soil. When you work with nature, when you work with microbial inoculants, the soil actually improves from year to year instead of becoming more and more depleted over time. Other microorganisms are superstars at making plant growth hormones, such as auxins and cytokinins. In fact, one microorganism feeds on exudates from the plant and changes it into IAA, indole acetic acid. That's the rooting hormone. Plants exude carbohydrates, remember? They feed the microorganisms. One of the carbohydrates they exude is called tryptophan. It's an amino acid. Well, some microorganisms literally snip off an ammonium ion, just one ion from tryptophan, changes it into indole acetic acid right on the surface of the roots, stimulating more lateral root growth and more root mass. Other microorganisms are better at making the cytokinins. Cytokinesis means cell division, so cytokinins are the hormones that stimulate cell division. So you combine the two in the dream team, those that make the auxins with those that fix the nitrogen, with those that make the cytokinins, put them together, it's synergistic. You'll get more lateral root growth, more root mass, better uptake of water and nutrients by the plant. Some of the microorganisms that colonize the root zone are also endophytes. They go into the plant. They penetrate the roots, go into the stems, into the leaves, into the buds, and process and produce biostimulants. They make phosphorus more available, they make nitrogen more available, and they even make growth hormones inside the plant. So it's not just the surface of the roots. It has an effect long-term on plant growth and health. Other plant growth promoting rhizobacteria are prolific enzyme producers. Remember, plants can't take up the large organic molecules, but microorganisms make digestive enzymes that break down the large molecules into a form the plant can take up. But they do more than that. If you find the right blend, you'll have those that feed each other while they're feeding the plant. For example, some microorganisms make the proteases that break down proteins into amino acids. That's a nitrogen source for the microbes. The other microorganisms are prolific at making cellulase. They break down the large molecules of cellulose into smaller sugar molecules. Combine the two, one's making nitrogen to feed the one that makes the sugars that feed the other. Those make sugars that feed the ones that unlock phosphorus. They provide phosphorus for the ones that fix nitrogen. You see, it's a team. It's not a single microorganism. It's a combination that feed each other while they're feeding the plant. Also, the byproducts of the digestive enzymes of the microorganisms have a direct effect on the plant. For example, the ones that make nitrogen available by producing amino acids. Amino acids aid with the uptake of calcium by the plant. Glutamic acid and glycine chelate the calcium and stimulate root cells to open calcium ion channels. The plant takes up more calcium. It strengthens the plant against fungal diseases on the leaf. So microorganisms in the roots make the plants more resistant to fungi like powdery mildew, bud rot, and other fungal attack on the leaves. So while microorganisms are feeding on carbohydrates, they make organic acids called Krebs cycle intermediates. They're byproducts of cellular respiration, cellular metabolism, and it has a direct effect on the plant. The Krebs cycle intermediates actually make some of the trace elements more available to the plant. They lower the pH in the root zone, sometimes by 2 or even 3 points, and solubilize minerals such as iron, copper, manganese, and zinc. Those trace elements activate enzymes that the microorganisms make and activate enzymes in the plant as well. If an enzyme is activated, even one molecule could do over 1,000 chemical reactions per second in the cell. Other select strains of plant growth promoting rhizobacteria, some of them are a little bit rare, but they're there in the environment, actually improve the photosynthesis process itself. The way they do that is by producing volatile organic compounds called VOCs. They're a bouquet of aromas that are very volatile that stimulate up to 600 different genes in the plant. One of those volatile organic compounds tells the plant, take up more iron. So they're making Krebs cycle intermediates that make the iron more soluble. They're making siderophores, which are very good chelators of iron, and then they're communicating with the plant. Even if they don't touch the plant, they're telling the plant, take up more iron. The plant takes up more iron, remember, makes more chlorophyll, harvests more light energy, makes more sugars, growth, reproduction, energy to fight off diseases, and also extra carbohydrates to leak from the roots to feed the microorganisms, to keep the process going. Another one of those volatile compounds also turn off the sugar signal inside the plant. That's really great for the finishing power of plants. When plants have enough stored carbohydrates to make it to the end of their life, they stop doing photosynthesis. They stop leaking carbohydrates and the microorganisms start to go dormant, go to sleep on the root zone. But microorganisms want to live, they want to reproduce, they want to have more colonies formed. So they make a volatile compound, shuts off the sugar signal so the plant doesn't know it has enough stored carbohydrates. So the plant will continue to do photosynthesis. It'll continue to bulk up and sweeten up and grow all the way to the day of harvest. Another benefit of the increased photosynthesis is even at the early stages of growth. If the plant makes more carbohydrates than it needs to use for energy to grow, it sends some of those extra carbohydrates to the roots. It's stored as starches in the root zone. Some of those starches are used for energy by the plant for active transport, to take up elements such as phosphorus and potassium. So if you have more stored carbohydrates in the roots, the plant will take up phosphorus faster. Phosphorus stimulates earlier flowering, more flowering sites. If there's more stored carbohydrates, you'll have a better uptake of potassium for carbohydrate metabolism to unlock the stored sugars so they can be shipped to the flowers and be stored in the fruits. So if during the vegetative growth stage you enhance the photosynthetic capacity, you'll have a faster transition from grow to bloom. In the lab, I've seen it. With high brix plants, with lots of stored energy, the transition was much more fast, much steeper than plants that were in the control group. In fact, in that early transition phase with the high brix plants, I had 20% more flowers in the early flowering stage. Finally, there are other select microorganisms, superstars of the rhizosphere, that are excellent at providing stress protection for the plant. They rescue the plants from stress. When a plant's under stress, heat stress or salt stress, drought stress, the plant produces ethylene. And ethylene downregulates the root growth and the shoot growth by the plant. But some microorganisms are ACC positive. They make an enzyme that breaks down the precursor molecule to ethylene. So it rescues the plant from stress and restores normal root and shoot growth.